Hello, Honors Biology students. In today's screencast, we are going to go over your virtual photosynthesis lab, my expectations, how the lab actually works, and where you are actually completing this assignment. So, if you go to Google Classroom, you're going to click on our class period. I'm going to click on period 2, but if you're in my 3, 6, or 8, it looks the same. So you guys know, go to the classwork tab. We're going to scroll down to our unit 2. And we're going to click on the Photosynthesis Virtual Lab. So here I've got the link. There's a couple little instructions here, but the main gist is that each of you is going to have your own virtual lab to actually type on. And then this video as well as the creating a line graph video are attached here. So any information or resources you need are going to be attached to this actual assignment. So let's take a look at the actual write-up. If we take a look at our write-up, it looks very similar to every other write-up that we've done so far. So when you're typing, please make sure you type in a different color. Red is fine, anything but yellow. So make sure you read your directions. My suggestion is before you actually click on the lab and do anything there, you come down to your procedures and you read through your procedures. Then go back, fill in your experimental design. You should have gotten a gist of what you're actually doing here. Do your purpose and your hypothesis, and then actually go collect your data. So let's take a look at what this lab actually looks like. So you're going to go here. It takes a minute to load. Once it loads, it should look like this. If for some reason when you click on the window it says like allow Adobe Flash, click allow, and then this should start to load and pop up. So this is the screen you should actually see. The important things you're going to need to know here are instructions. So you can read through this. It kind of points to what is going on here. And the start button. You're also going to need to know the slider here that moves our light source closer and farther away from our Elodea. We've seen this plant before. This is the same seaweed, our Elodea, that we looked at under the microscope when we were looking at plant cells. So if we take a look back at our procedures, again, you can read through these guys, but I'm going to give you kind of a quick overview of what you're doing during the actual experiment. So if you take a look at your table, you're going to have various distances from the light. So we're going to start at 200 centimeters. So if I click back over to the lab, I'm going to move the light source all the way back. So if you look at the bottom, you can actually see where it says 200. And then I'm going to click Start. So this brings me to this window. The first thing I want to do is click over on this guy to counter. If you take a look at the screen here, this is the Elodea plant. It's really close up. And what we're actually counting are the bubbles that are coming off the Elodea. It's going to be your job to figure out what gas is inside of these bubbles. But what we want to do is count the number of bubbles. Right now, my bubbles are going pretty slow, so it's pretty easy to keep track. But as the bubbles start to speed up, and you'll see what I mean when you actually run the experiment, this counter comes in handy. So again, if we take a look at our table, it wants us to count the number of bubbles that come off our Elodea in 30 seconds. So if I click reset, it brings the red second hand back to 12, and then we can start counting until it gets to the 6. So I'm going to click reset and start counting right now. So that's 1 bubble, 2 bubbles, 3 bubbles, 4 bubbles. You get the idea. I'm not going to do it for the whole 30 seconds. But you're going to count your total number of bubbles in 30 seconds. Once you get that number, you're going to come back here to your data table, and let's say that if I continued counting, I ended up with 10 bubbles in 30 seconds. Then it wants me to figure out the number of bubbles in 60 seconds. So all I'm going to do is take this number that I got for my 30 seconds and multiply it by 2. So in this case, 10 times 2 would be 20. Once I finish with that, I'm going to go back here. I've collected all of my data for that distance, so I'm going to click back. Once I go back, then I'm going to adjust my light source. My next value here was 180 centimeters. So I'm going to move my light source up, click Start, and count the bubbles again. Again, don't forget to switch to the counter. Okay? So you are going to do that for all of your values here. So once you've collected all of your data, then you're going to graph. You're going to graph using the exact same method we did for the potato core lab. Your instructions here are correct. Don't worry, I've checked. Again, if you need that refresher, go back to the video that's on Google Classroom and watch it just to make sure you are on the right track. Once you have created your graph in Excel, you're going to copy and paste it here, again, just like we did for the potato core lab, and then you're going to move on to your discussion. Your discussion includes your conclusion. Again, make sure you're meeting all of these criteria. Restate your purpose, restate your hypothesis. That can come directly from the top. Support or reject your hypothesis, draw a conclusion, and support it with evidence and data. Remember, data means actual numbers. Then your explanation is going to be the science behind your results. So why did we get the results according to the concepts we've learned in class? I want you to think about that photosynthesis equation. What's going in? What has to be coming out? Hint. The bubble is filled with gas. What gas comes out at the end of photosynthesis? Okay? So this virtual lab is going to be due on Monday. You're going to have Wednesday in class and Friday in class to actually work on this. You should be able to finish the majority of this in class and have very little homework to do outside of class. If you have any major questions as you're working through, feel free to either shoot me an email or send me a remind message. I will get back to you quickly. If there are any other major questions, your classmates are there, they can help you. And like I said, email is always welcome. Have a great rest of your week. I will see you guys on Monday. Please be good for your sub, and good luck on the lab.